And like most of the hormones that we've been discussing, the history of hormones starts off with some what might be considered to be bizarre or unusual observations. One of the first observations that has been attributed to ethylene, but was not known to be attributed to ethylene at the time, was the fact that pineapples can be induced to flower by smoke. So you make a fire and let the smoke from the fire blow past the pineapples. It induces them to flower even at times when they normally would not flower. Okay? So there's some component of the smoke that is inducing the pineapples to bloom. And long before farmers, particularly in Hawaii, knew that this had anything to do with ethylene, they used that observation to synchronize the flowering of the pineapple plants. So rather than having the pineapple sort of blooming over, some physiological range of time, depending upon environmental conditions, get them to all bloom at once. It makes it easier for picking. Yeah.
Is this traditional agriculture in pineapple agriculture in Hawaii, or more modern? How far back does this go?
This goes back a ways. Goes back before the 1930s when ethylene was first discovered. So I don't know exactly what the timing of it is. I don't know when pineapple agriculture became sufficiently strong in Hawaii that someone could accidentally have started a fire and got all their pineapples to start blooming simultaneously. But it was before 1930s. Okay, it preceded that. One of the ones that was very commonly observed is once street lamps in places like England were run off of natural gas. One of the things that was observed was that trees that were in close proximity to the street lamps, the leaves would senesce pretty early. Okay, so that there was some component in the natural gas that was being used in the street lamps that was inducing leaves to senesce and fall off early, right? So it's not necessarily a very something that might make you concerned about the characteristics of whatever was in the gas they were burning. The sort of even scarier one is the idea that's referred to as the triple response. This was observed by a Russian plant physiologist who was growing pea seedlings in the dark in the basement of his Russian apartment. And what he observed was when he grew them in his basement, that there were three responses. There were shorter hypocotyls, and thicker hypocotyls, and also they tend to be less responsive to gravity. So this so-called triple response, here's a picture of triple response in peas and in Arabidopsis. So you can see very clearly in the peas shorter and thicker hypocotyls. And it's less clear in these pictures the loss of the responsiveness to gravity, but it's certainly one of the features of it. And what Nelgerbo, the guy who discovered this, found was if he opened the window and let fresh air into his basement, then the seedlings grew normally. Okay, so there was something in his basement air, in the laboratory where he was working, that was causing this response. And when he let fresh air in, this response went away. Once ethylene was discovered, that it could induce some of these physiological responses, like the triple response, it very quickly became apparent that a lot of the different ethylene responses seemed to be related to auxin, particularly, so if you take exogenous auxin, apply auxin in a paste to a plant. One of the things that happened was this induced ethylene synthesis. So for a long time, it was assumed that ethylene was nothing more than a signaling molecule that's part of the auxin signal transduction pathway. Right? That it's not a signaling molecule unto itself. But we now know that the conditions under which this auxin was applied, when it was sufficient to induce ethylene synthesis, with a very few exceptions, is non-physiological. That is, there are a few conditions where auxin can cause ethylene biosynthesis. We'll see when we talk about the biosynthetic pathways. One of the key enzymes can be turned on by high auxin. The question is, how often does the auxin concentration get high enough to actually do that in the plant? And the answer is not very often. So the auxin-induced ethylene biosynthesis is largely non-physiological. So if you go back and look at plant physiology textbooks even 20 years ago, almost everything to do with ethylene was induced by auxin. That's wrong. There are a few things to do with ethylene that are induced by auxin, but most are independent of auxin. Okay. So ethylene is actually kind of a bizarre molecule to be a hormone. It's a pretty simple molecule, right? Are there any other hormones that are as simple as ethylene? Any other plant hormones? No. How about animal hormones? Don't know your mammalian physiology? There's one mammalian hormone that's even simpler than this. Nitric oxide. That's a pretty simple molecule, right? Okay. But because ethylene is a simple molecule, it has physical and chemical properties that distinguish it from all the other plant hormones that we're going to be talking about. So one of the key ones that we have to deal with, it's a gas. It's volatile. The equilibrium between it being in solution and being in the air favors it being in the air. So anytime ethylene is produced, it's going to be produced in the cytoplasm of the cell. It's going to be in water. But the equilibrium is for that ethylene to go out of the water into whatever air it can. Right? And if you think about it, most plant tissues, the living part that would produce the ethylene is pretty close to the air. You know, whether you're talking about leaves or stems, there's not a long diffusion distance there. So the fact that it's gaseous, the fact that it's volatile, is going to play a really important role in thinking about what ethylene can do. It does have moderate solubility in water. It might be pretty surprising that it's water soluble because it's pretty much a hydrocarbon. What makes it water soluble at all is it's a small hydrocarbon. Right? The double bond contributes a little bit, but mostly it's the size of the water molecule. Obviously, water solubility is essential if it's going to function biologically, because where it's produced and where it acts are almost certainly going to be in water. Right? So that water solubility is important. The other thing is it's very membrane permeable. No transporters needed to get ethylene through the membrane. Okay. So this has some important outcomes as far as ethylene function in plants are concerned. If it's gaseous and if it's membrane permeable, can you ever keep ethylene in a cell? No. It's going to diffuse through the membrane, and if that diffusing through the membrane lets that lets the ethylene come in contact at a air-water interface, it's going to go into the air pretty quickly. So that means losses of ethylene from cells is going to be something that's very different than any of the other hormones that we're talking about. Because none of the other hormones, we've talked about a number that are membrane permeable. Right? That means they can get in and out of cells easily. But none of them are volatile. None of them are gaseous. All right, so that's going to play big differences. And so one of the things that you should be thinking about throughout our discussion and your review of ethylene functions is that remember we talked about what are the properties that or what are the processes that control the local concentration of a hormone. Synthesis and degradation, transport in and transport out, conjugation, deconjugation. That picture is going to be very different for ethylene than it is for any of the other hormones. So what does that mean about ethylene transport in plants? Just knowing this, what does that tell you about ethylene transport in plants?
Probably goes up.
It goes up? Why do you say that? Okay.
But I don't see how it could
Up like in the xylem? Couldn't it go down in the phloem?
in the air.
Oh, it goes up in the air. Okay. So is movement of ethylene in the air, would we want to consider that to be transport within a plant? What, you mean even if it could get it from the roots to the leaves, what's the downside of that? What's the downside of transport from one part of the plant to the other part of the plant through the air? There's no way to control where it's going to go, right? And how about on a day like this? It's not going to go where you want it to go, right? It's going to get blown away, right? So transport in the air, basically we should think of any time when the ethylene gets into the air, it's gone, right? It would only be chance if it got to another part of the plant that it needed to get to, okay? So transport within the plant has to be the rule. But what does this tell you about transport in the plant? Stella? Okay. Why do you say that? Okay. So does that mean ethylene will diffuse from the roots to the leaves? Okay, so it could diffuse through the cells. Could it diffuse through the cells from the roots to the leaves? It would take a very long time. Okay, so that right away tells us it's probably not going to happen. But what other characteristics are we looking at here that tells us it's unlikely that ethylene would move by diffusion through cells from the roots to the leaves? What's going to happen to ethylene along that path? Yeah, it can cross membranes, it'll get to an air-water interface, and it'll volatilize and it'll be gone. Okay? So what's the likelihood of long-distance transport of ethylene in plants? Not very great. So what does that mean about what's going to be the key things that controls the concentration of ethylene at any part in the plant? Either storage forms, right? Or what else?
Well, it's it could be transport. You have transport protein or synthesis.
Well, if it's a transport protein, it doesn't help very much if it's volatile.
But you can get an activated form.
Yes, if you get a conjugated form or something like that, yeah. So it's either going to be transported as conjugated form, or if it's not, then what's going to determine is the local chemistry, synthesis, degradation, or conjugation, deconjugation, right? And that's it. You have just figured out all the key factors associated with ethylene, controlling local ethylene concentration. There are a few cases where ethylene can be transported as a conjugate, or we'll see actually a precursor. But in virtually all other cases, the controlling factors are local synthesis. And how about degradation? Do we need to be concerned about mutants that alter ethylene degradation. So remember when we talked about cytokinin, so cytokinin, if you have a cytokinin degradation mutant in the shoot apical meristem, it makes a very small meristem. If you have a cytokinin degradation mutant in a root apical meristem, it made it much bigger. Would you see similar effects with ethylene degradation mutants that you do for cytokinin?
Maybe not.
Why not?
Because maybe just the fact that it volatilizes is enough to get rid of it.
That's right. So the natural losses are sufficient that degradation, changing the amount of degradation, has virtually no effect.
So what kind of thing degrades ethylene? Is there something that degrades?
Are there enzymes that degrade ethylene? Remember from the text?
Weren't there just enzymes that like degraded the, what do they call it, the precursors?
There are enzymes that can deal with the precursors. There are no enzymes that are there specifically to degrade ethylene. So you can degrade ethylene to ethylene oxide, you can degrade ethylene to glycol, but there are other enzymes for which ethylene is a secondary, poorer substrate. There are no enzymes that are directly involved in ethylene breakdown to deal, to control local concentrations of ethylene. And it makes sense that there aren't, because the normal losses, because of membrane permeability and volatility, mean that anywhere that ethylene is being produced, it's not going to get very far. It can't diffuse very far before that diffusion gets it into the air and it's gone. Right? So we need to think about this in a very different perspective than what we've talked about for the other hormones.
Reporting it in the gas form outside of the plant, it ever plays a physiologically important role, since we think of like methyl jasmonate, methyl salicylate as potentially having control.
Yep. Yeah, so there are circumstances where we've talked about volatile compounds that can give signals from one plant to another, or even within a plant. So jasmonates and methyl salicylates are good examples of that. What must be the difference? Why would those compounds be able to induce a response at a neighboring plant and ethylene not be able to produce a response in the same plant or a neighboring plant, assuming that the transport in the air, either diffusion or moving in the wind, is the same for those two? And they will be. They're all small molecules. Yeah, presumably it must have something to do with the sensitivity of the receptors, right? That for the jasmonates and the salicylates, there must be greater sensitivity that can detect lower levels. I don't know for sure that that's the answer, but that would be the logical expectation based on the fact that once these compounds get into the air, they're going to move wherever the air is going, right? And so the concentration is going to decrease rapidly as you move from the source to anywhere else. Okay? All right, so it's really important that you be able to use these three specific characteristics of ethylene to be able to think about ethylene function in plants. Okay, so biosynthesis of ethylene is an interesting pathway, and I won't go into the details of it, but we'll talk a little bit about it. The compound that's used in the synthesis of ethylene is a very important compound in all organisms. It's S-adenosylmethionine. And the normal role that S-adenosylmethionine, who knows what it is? Who's the biochemist in here? What's this normally involved with?
I would have to guess in DNA, but
Well, it's certainly related to DNA because it's got the riboside here, right? But it's something else.
Amino acid,
It is involved in some amino acid, but the fact that it's methionine, it is an amino acid already, right?
I don't remember that yesterday.
No, you don't remember?
it's
It's this guy right here. S-adenosylmethionine is a methyl group donor. So whenever you want to methylate something, whenever you want to add a CH3 to something, almost always it's S-adenosylmethionine. So this bond, you notice that there's a sulfur here that's got 3 different carbons bound to it. That's not stable. Right? That's not stable. That's not a normal electronic configuration for sulfur. It wants to give up one of those guys. And under normal circumstances, it gets rid of this group. It donates this group to some other molecule. But in the synthesis of ethylene, it is this bond that breaks. So this 4-carbon nitrogen-containing compound is what's broken off. And even more bizarre is that the product of that forms a 3-membered carbon ring. This ACC, aminocyclopropane 1-carbonic acid, carboxylic acid, is a very unusual compound. You would think, for example, the fact that it's got 3 carbons in a ring, that would be a highly strained ring, right? Carbons don't like to form bonds at those angles. And it's true that they don't. But surprisingly, this is a relatively stable compound. This compound can be transported in plants over long distances. It's not volatile. It's not membrane permeable because it's charged. Right? So it will stay in the cells you put it in. Right? If there's transporters that will move this in and out of the cell, you can move it around in the direction you want to. So ACC has all the normal characteristics that we would think of as a hormone that can be moved around. But there are relatively few circumstances in the plant where that happens. Under most circumstances, the ACC is relatively rapidly converted into ethylene with some interesting byproducts. Carbon dioxide. What's this? HCN. Hydrogen cyanide. Remember what hydrogen cyanide does? There are some cyanogen compounds that produce hydrogen cyanide as a product, but so what? Why do we worry about hydrogen cyanide? Yeah, it inhibits. It's a very effective inhibitor at low concentrations of the last step in respiratory electron transport. It inhibits the cytochrome oxidase. Right? So that basically blocks most of the ATP production in cells. Right? So producing much of this can be a real problem. So the fact that CO2 and hydrogen cyanide are products, and that the conversion of ACC into ethylene also requires an input of oxygen, turns out to have some very interesting physiological characteristics in terms of the way plants take advantage of this, but it also has some very important physiological characteristics if you are a post-harvest physiologist, because one of the things that we know that ethylene plays an important role in is fruit ripening. And so if you want to store fruit and not have it ripen, understanding the pathway of ethylene biosynthesis is going to be an important thing. Okay. So one other aspect of this that we should mention is that sulfur is an important element in plants. It's involved in a number of different types of amino acids. So you can't just make ethylene and have this sulfur be lost. There has to be some mechanism of recycling compounds that contain this sulfur back into normal metabolism. Okay, so this Yang cycle is a series of reactions that take the 5-methylthioadenosine and recycle this back into methionine again. So a number of interesting things that are going on here. So one of the questions we should be asking ourselves are, what are the reactions in this sequence that limit the synthesis of ethylene? And under most circumstances, It is this enzyme ACC synthase that converts S-adenosylmethionine into ACC. So when ethylene synthesis is turned on in specific cells or specific tissues, the enzyme that typically changes its concentration the most is ACC synthase. There's sufficient ACC oxidase present in most tissues to account for the rates of ethylene biosynthesis. Okay, so that gives us an important clue that signal transduction pathways that regulate ethylene biosynthesis, the end product that's being regulated should be ACC synthase. Okay. There are a couple conditions, fruit ripening in particular, where ACC oxidase is also upregulated, and we'll see why it's different for fruit ripening in just a minute. But in most cases, it's ACC synthase that's controlling the rate of ethylene biosynthesis. Okay, your book talks about inhibitors of these processes, and as I mentioned before, if you're a post-harvest physiologist, inhibitors of these processes might be very important in keeping fruits from ripening. So a couple of the inhibitors, you don't have to know what these compounds are, AOA or AVG. They are not inhibitors that are specific to ethylene biosynthesis. These are inhibitors of a function of a compound that's involved as vitamin B6. It's pyridoxal phosphate. Pyridoxal phosphate is involved in a lot of amino transfers. So the enzymes that move amino groups around between different types of amino acids, you notice that there is an amino group that's involved in these reactions here. So these compounds here inhibit the function of pyridoxal phosphate. Pyridoxal phosphates, if you inhibit that in humans, it causes serious biochemical problems. It would behave like a vitamin B6 deficiency. Okay? So we're not going to use these inhibitors to block fruit ripening when we store apples, right? Because it would cause some serious problems. But one of the things that we can easily look at to block ethylene biosynthesis associated with fruit ripening are the fact that oxygen is required in this last step. So low oxygen would be a very useful thing to use in storing fruit. What's the downside of using low oxygen concentrations to store apples in, for example? Could we store apples in a zero oxygen environment? They'd stop respiring. Something would happen before they die. Our apples, we pick the apple off the tree, right? And we put it in storage. Is that apple alive? Are the cells still alive in there? Absolutely. Are they respiring? Where are they getting their energy for respiration? Sugars and stuff that are stored in the apple, okay? So if we take the apple and we put it in zero oxygen, what's going to happen? Will the apple die? It'll go anaerobic, and then what's going to happen? Fermentation. Is that bad for the apple? Well, yeah, okay, so if you're trying to make Applejack or something, maybe it's not so bad. But if you're trying to sell the apples as something that you or I might eat, why is fermentation bad? Yeah, children won't be allowed to eat them. Okay, yeah. We're missing one important point from respiratory physiology. What's the desirable part of an apple as far as humans are concerned? Yeah, sugar, right? That's why apples were so important 200 or 300 years ago in the U.S. because there were no other good sources of sugar. There was sugar cane, but you couldn't grow it very many places. Sugar beets and apples were basically the only sources. What happens to the amount of sugar in an apple if it switches from aerobic to anaerobic respiration? What happens to the rate of sugar utilization in the apple for the cells to maintain their normal biochemistry? Goes way up by about a factor of 20 or so, right? So why don't you want to store apples under anaerobic conditions? Yeah, they won't be sweet. You'll use up all the sugars in them, right? So if you don't lower the oxygen concentration low enough that the apples start to ferment, that they go anaerobic. But you can lower it enough that you prevent the function of this enzyme ACC oxidase. So here's a good example of how understanding the biochemical pathway has real practical results for agriculture. That if we understand the role that oxygen is playing, both in respiration and in ethylene biosynthesis, then we can design logical conditions under which apple storage can be accomplished. And we're getting pretty good at it. I mean, if you buy apples in July, well, maybe they came from New Zealand, but you get a lot of locally grown apples around here in July. And for the most part, they're okay, right? If you just had a normal apple in July, what would it look like? What would a normal apple look like in July? Yeah, right? So what is happening in the ripening process? Softening of the cell walls. Yeah, so remember when we were talking about cell wall characteristics? What is it that changes between what we would consider to be an edible apple and an inedible apple? What changes there? Yeah, it's the pectins, right? So the pectins are their stuff in the middle lamella. It's the glue that holds the cell together. So when the cells are glued together, when you bite into it, the cells rupture because the mechanical force you're imposing with your teeth is not strong enough to break the bonds between the cells, to break up the connections between the cells, but is strong enough to break the cells themselves, right? Once that fruit ripens, once those pectinases are produced, that middle lamella is gone. And now when you bite into it, the cells just slide past each other. It tastes mushy. All right? So remember, what ethylene is doing is promoting that ripening process. Why does ripening happen at all? From a purely physiological perspective, why does ripening happen? Why do fruits ripen? Why do fruits ripen?
Well, usually fruits are for seed dispersal.
Yep.
So these kind of like fleshy fruits basically usually are eaten by animals.
Yep.
And so it's not good for the plant if the animal eats the fruit before the seed is mature.
Right. So in other words, the seed maturation process has to be timed correctly with what's happening with the ripening of the fruit, so that the ripening of the fruit, really what the ripening of the fruit is signals to animals, come eat me, right? Right? So do deer eat apples at the same stage that we eat them? Do deer like those nice, crisp, unripe apples? No, they like the mushy things that are all turning brown. And yeah. Why? What's changed? What changed in that apple? It got ripe, but as far as the deer is concerned, sugar might be part of it, but sugar's in the nice crisp apple too, right? That's why we eat it.
It's easier to eat it, and also it fell off the tree.
Okay, fell off the trees. So we don't have to have giraffes to get to the apples. But there's something else, and you know what it is. You're just not thinking about it. If I brought you into a room and I had two piles of apples, one were the nice crisp apples that we would normally eat, and one are the apples that are nice and ripe that the deer would like to eat, could you tell the difference between those two apples with your eyes closed?
Yes.
Smell, right? So there are compounds that are produced as the result of the ripening process that are attracting the deer to eat them, right? And the fact that they're mushy and stuff like that just makes it easier for the deer to get to eat everything, to get to the seeds, right? Because the seed dispersal is counting on the fact that the apple gets eaten by the deer moves somewhere else, defecates, and the seeds get dispersed that way, right? So you got to be very careful about remembering from an evolutionary perspective what ripening is all about, and from a human perspective what ripening is all about. So when we think about tomatoes, for example, we want to have tomatoes at a very specific stage of ripening, right? We want them to ripen up to the point where they're nice and red and fleshy, but we don't want them to turn to mush, right? Right? Same with apples. There's a whole sequence of events, and we'll talk about in just a minute the role that ethylene plays in that ripening process. Okay, so because ethylene is so important in fruit ripening, so for example, how many of you like avocados? Right? So when you go to the store, usually, this year's actually been better than normal, but usually when you go to the store, the avocados are closer to hand grenades than they are to anything that's edible, right? So what do you do? Put it in a paper bag. Put it next to your bananas if you really want them to ripen fast, right? So production of ethylene by one fruit can ripen other fruits. This is one of the ways that ethylene was discovered, that transports of different types of fruits, when one fruit ripened, basically everything ripened. Okay? So in that sense, that might be bad. But in another sense, triggering ripening of fruit that aren't ripe might also be good. And one of the ways this is done is by using a compound that in water naturally degrades to produce ethylene. And the commercial name for this compound is ethifon. So ethifon is a compound that breaks down in water slowly. It's not a fast reaction. Breaks down slowly in water to release ethylene. And it's commonly used to trigger ethylene-dependent processes, in particular, ripening of certain types of fruits. Right. So clearly, we've been selecting fruits for certain characteristics in terms of their ability to be transported and their ability to store. And, avocados that you can pick like hand grenades, transport, and 2 weeks later have them get ripe. So we've been selecting for those for a while. I don't think ethifon is used very commonly to ripen avocados commercially. But it certainly is used for synchronizing ethylene-dependent processes, particularly, for example, plants whose flowering is ethylene-dependent.
So how do they slow down fruit ripening? It's not oxygen.
Well, oxygen would do it. Lowering oxygen, but not making the cells be anaerobic. Low oxygen is one. And if we go back to this figure, what other information on here would be useful in thinking about conditions that would inhibit ethylene production?
You would just increase the product, right? Because if you increase CO2, then it's going to...
Yeah, so increasing CO2 is the other part of this. Since CO2 is a product of the ACC oxidase reaction that produces ethylene, high CO2 will just, by Le Chatelier's principle, will slow down this reaction as well. So those are the two main factors that are involved in apple storage. High CO2, typically 2 to 5% CO2, and low oxygen. And low oxygen for this for inhibiting this reaction is on the order of 0.5%. So normal oxygen is 21% in the atmosphere. And the Km for oxygen in respiration, anybody know what that is? It's really low. It's like 0.01%. Right? So you can lower oxygen a lot and not inhibit respiration. So you go down to 0.2, 0.1% oxygen, and respiration's going along just fine.
Why do we suffocate so easily?
Why do we suffocate so easily? Good question. You're all physiologists. Why do we suffocate so easily in 1% when oxygen gets lower than normal?
Carbon dioxide, or does that have to do with it?
Carbon dioxide has something to do with it, but low oxygen is the main thing. So if I decrease the oxygen concentration in this room to 1%, you'd all be unconscious pretty quickly. Me too. We'd all be unconscious pretty quickly. I don't want to exclude myself from humanity. But why? You're a physiologist. Why?
Transport.
Yeah, basically. So the diffusion of oxygen across membranes requires a certain oxygen gradient, right? If we lower the oxygen concentration in the air by a factor of 10, we've lowered that gradient by a factor of 10, right? So, I mean, this is what this class is all about. These are the sorts of things that you should be able to think naturally about. If we're talking about why lowering an oxygen concentration has an effect on an organism, you say, okay, well, what does oxygen do? What are the processes that get oxygen in and out? You should right away be thinking about diffusion across interfaces or membranes.
But freshman year we went on a tour to the orchard.
Yep.
And we were in the place where they store the apples, and they were explaining us like low oxygen, high CO2, and we were inside the room
Yep.
for like many minutes, and
Yep.
nothing happened to us. So
Yeah, so many minutes is probably, it depends on how long many minutes is.
That room was off,
Yeah.
too.
I mean, I can almost assure you that
Oh,
if that room, if you were in there with the door closed, under the normal conditions, I would think that you wouldn't last more than 30 seconds or a minute. Yeah.
So maybe it was off. It was for sure off.
Yeah,
We went with Ian.
Yeah, so obviously lowering the temperatures as well is going to have an effect. Okay, so we're spending a lot of time on thinking about practical aspects of this, which is good because that's applying the physiology to things that we think are important. All right, let's spend a few minutes talking about how we can use mutants to sort out ethylene signaling in plants. And most of your questions this morning were related to understanding ethylene mutants. And I want to emphasize how you go about thinking about all of these things we've talked about. If your perspective is to sort of, memorize everything that's going on in these different types of mutations and for all these different hormones, okay, that's one way to do it. But it's not a very practical way to do it. The way to do it is to remember that every single step in a signal transduction pathway, whether you're talking about the receptor, a signaling molecule in the middle, or the response protein at the end, every single step has two states, on and off. And that on-off can be regulated in one of two ways. Negative regulation by the previous step or positive regulation by the previous step. That means every mutation that can happen in signal transduction pathways will fit into one of four categories. Negative mutation stuck on, negative regulation stuck on, negative regulation stuck off. Positive regulation stuck on, positive regulation stuck off. That's it. Those are the only four possibilities. Now, obviously, when you get multiple receptors and things like that, when we talk about ethylene, it complicates it a little bit. But don't treat these things as being separate from each other. The common thing that makes all this palatable, understandable, is that signal transduction pathways have more in common than they have not in common. Understand what those common things are. Okay? So let's put this to good use right now. Here are two different types of ethylene mutants. So first of all, let's not worry about where the mutation is, but describe to me the phenotype of this mutant. It's tall. And is this grown in the presence or absence of ethylene? How do you know that? Yeah, so all these guys are showing triple response, and this guy isn't. So in a general sense, how do we categorize this mutation? Ethylene insensitive, right? So in the presence of ethylene, all the normal plants are showing the triple response. This guy looks normal. right? So this is an ethylene insensitive mutation. In the presence or absence of ethylene, the plant appears normal phenotype. How about this guy? How do we describe this mutation? Constitutive in what sense? It's responding to ethylene even when it's not there. So this would be, the signaling pathway would be constitutively on. So I'll give you a little challenge. You should be able to go home and tell me, of the 4 categories of mutations, how could we account for these 2 different phenotypes? Right? And I don't care whether, you don't close your eyes. You don't need to know anything about the components of the ethylene signal transduction pathway. Just tell me in the most general sense, for those 4 different types of mutations that we just talked about, which ones could account for these. Once you do that, then you can go back and look at the characteristics of the mutations that are described in the textbook and see how it fits together. That's what I want you to be able to do. It's not look at the mutation and immediately understand what's going on. I don't expect you to be able to do that. But if you compare it with those 4 different categories, positive regulation stuck on, positive regulation stuck off, negative regulation stuck on, negative regulation stuck off, you should be able to describe mutations that can account for these phenotypes. You should be able to compare mutations between 2 different types of mutants in the same organism, in the same pathway, or 2 different types of mutations in different signaling pathways. It's the understanding of how signaling works that's important here. If you work on analysis of mutants, that's how you're going to do it. You understand how a signaling pathway works, you look at the phenotype of your mutant, and you see that's consistent with this type of mutation and not with these other types of mutations. Okay? All right, so let's think about this in the context of ethylene. So one of the things that is different about ethylene, what's the thing that's most different about ethylene compared to the other signal transduction pathways that we've talked about? Okay, so the receptor is a negative regulator. The other pathways we talked about so far, auxin, gibberellins, phytochrome, it's at the other end, in the response end, where the negative regulator is. Here, the negative regulator is at the receptor end. One other important difference. Redundant receptors, right? So it's not just one ethylene receptor. There are, in Arabidopsis, there are 5 redundant receptors. So we need to take both of those into account when we think about how we explain these different phenotypes. Okay, so the normal state of ethylene receptors in a plant in the absence of ethylene is, would we categorize these receptors as being on or off? In the absence of ethylene, would we categorize these receptors to be on or off? On, right? What is their on function? Repression of the next step, turning off negative regulation of the next step. So in the absence of ethylene, these receptors are on, and that on function is to turn off. The negative regulation is to turn off that next step.
So normally they're
These ethylene receptors are negative regulators. And remember that every step in the pathway is going to have an on and off state, right? In this case, in the absence of the signal, the regulator is on. If this was a positive regulator, what would the state of the receptor be in the absence of the signal? Off, right? So those are the distinctions that you need to be able to make. So in this case, in the absence of the signal, these are on, and that on function is to turn off the next step. That's the negative regulation.
What would you then think about after
Say that again?
Can you think of ethylene then as a positive regulator of all the pathways? Because, when ethylene binds to the receptor,
Yeah.
it turns it off.
Yeah, and that wouldn't, yeah, and that would really be true for all signals, right? All signals, in their absence, something isn't happening, and in their presence it does happen. Yeah, I guess. I mean, yeah, so this is, the semantics of this can get messy. But
I'm just trying to kind of put the ethylene in the categories that you said, but now I'm really confused. Because in reality, ethylene turns on the ethylene response pathway.
ethylene turns on the response pathway by turning off its receptor, right? The receptor is on normally in the absence of ethylene, and that on function is to turn off the next. Okay, so now let's think about what happens then if ethylene binds. So if ethylene binds, it turns off the receptor. And turning off that receptor releases the inhibition of the subsequent step. Don't treat this as any different than what we talked about, for example, the AUXIAA protein in auxin or the DELA protein in gibberellins. They're doing exactly the same thing. They're just at the other end of the signal transduction pathway. We're not degrading the receptor, but we're turning off its function. Right? So the presence of the upstream signal, in this case, it is the signal, it's ethylene. The presence of ethylene turns off the repression of these proteins. Right? Remember back in the very first chapter when we talked about signal transduction, that plants use inactivation of negative regulators. The inactivation is happening as the result of ethylene binding to these receptors. It's inactivating the inhibition of the next step. So when these receptors bind ethylene, the next step is turned on. Okay? So the distinction between these two states, we okay with that? Please make sure you understand it. Good question. Does ethylene have to bind to all the receptors in order for the response pathway to be turned on? What do you think? If ethylene was bound to these four but not to this one, what would this one be doing? It'd be inhibiting the next step, right? So in this model, does ethylene need to bind to all the receptors in order to turn on the pathway? Yes, right? So if we think about this case, where we make one of these receptors insensitive to ethylene, we remove its ethylene binding site, but keep all its other functions normal. What's the phenotype of this mutant going to be? Don't look at the picture, use your brain. Describe what the phenotype of this mutant is going to be in terms of the ethylene signaling pathway. Is the ethylene signaling pathway going to be on or off in this mutant? Always off, right? Because no matter whether ethylene is present or absent, this receptor is going to shut off the rest of the pathway. Right? So this is a mutant that would not be able to respond to the presence of ethylene. This would account for the skinny tall guy in the presence of all the triple response. Right? So what does this mean about what can we tell from a mutant that shows this phenotype? Can we say where the mutation is? Can we say the mutation is in the ethylene receptor? If you see this phenotype, can we say the mutation is the fact that it's behaving as if ethylene isn't there even when it is? Not necessarily, right? That's right. Yep, it could be anywhere in the response pathway where, in the presence of ethylene, the response pathway stays off. That's the only thing it tells you, right? Let's just say that we knew the mutation was in the receptor. Does it tell you what gene the mutation is in? No, because there's 5 redundant genes, right? So think about this from the molecular biologist who is analyzing phenotypes of mutants and trying to figure this out. Think it might be a little confusing to sort out what's going on here if you have 5 redundant receptors? It wasn't until the Arabidopsis genome was sequenced and it was clear that there were 5 redundant receptors that our understanding of this sort of fell into place. The gene jockeys who were trying to sort this out before we had the Arabidopsis sequence were doing a lot of this. Yeah, so basically the phenotypes that have been looked at, a mutation that blocks ethylene response in any one of those will give you this phenotype. Okay? Yeah, so it's a perfectly legitimate question. Why the redundancy? Why the redundancy? I don't expect you to be able to answer it. I'm not sure the experts can answer it. Right? There's lots of things that happen in evolution that aren't necessarily happening for a reason as long as it's not negative, as long as it's not bad for the organism. Right? What's the... I believe the textbook mentions something about changes in sensitivity to auxin with numbers of functional receptors. Sorry, not auxin, ethylene. What happens to the sensitivity to ethylene if we, instead of having 5 receptors, we only had 1 receptor? Is it more sensitive, less sensitive, or the same?
Do you mean kind of receptor or
No.
number of receptors?
Nope. Let's make it easy. Let's just say there's one type of receptor. Okay, just one type of receptor. And we're going to imagine a cell with 1,000 ethylene receptors in it of the same type, or a cell with 1 ethylene receptor. And they are this negative response type. Which plant is more sensitive to ethylene? The one with 1,000 receptors or the one with the 1 receptor? Okay, why? Why the 1 receptor? It only needs to bind to the 1 receptor to do what? Right, because that's the only one that's holding the pathway back, right? Where the one with 1,000, you've got to bind 1,000 ethylenes before you can turn the pathway. Because if you have 999 that have bound ethylene and 1 that hasn't, the pathway's off, right? So make sure that you understand that.
But is there a difference? Say if there's 10 receptors of one kind and 10 receptors of another kind compared to 20 receptors of one kind?
Yeah, so what would be the important thing we would need to consider if we're talking about different types, if we have 5 different types of ethylene receptors? What would we need to consider? Binding affinity, right? What's the difference in their binding affinity? And I don't know what the difference is between these guys. Almost certainly there's some difference. Whether it's large enough to contribute to what you're saying, I don't know the answer to that. Yeah.
But if you have multiple receptors compared to a single receptor, wouldn't there also be a difference in terms of the concentration and the amount of the components of the signaling pathway? Like if you had, there's only the receptor would be, well, the negative regulator can only turn off so many components of the signal transduction pathway, right? It must have a saturation point.
Yeah, that's true.
Yeah.
That's true.
So when you have more receptors, it means that you could possibly have a larger number of proteins involved in
Yes.
involved in the signal transduction pathway. So when you had those negative regulators turned off, you have more of a response.
Yep. So obviously it's more complicated than the simple picture that we just talked about. I mean, you need to think about it in terms of how does binding of a signaling molecule change the concentration of free molecules that can bind to other receptors. You need to think about the stoichiometry of receptors compared to the next step in the pathway that the receptor is controlling. All of those things are going to be important, right? But in the simple approximation, you correctly determined that you would expect that in this sort of situation, decreasing the number of receptors should actually increase the sensitivity to ethylene. Okay? All right, let's do another one. So these are mutations in which they're still capable of binding ethylene, but whether or not they bind ethylene, their negative regulation of the next step is gone. So what's the phenotype in terms of the signal transduction pathway for this type of mutant, Anna? Yeah, it'll always be on. It'll behave as if ethylene is there even when it's not, right? And one of the things the book talks about, it's mentioned up here in the label, is that in order to see this sort of phenotype, you actually don't have to mutate all of them. You just need to mutate most of them, right? So this is maybe getting at the sort of things that Grace was talking about. The stoichiometry of the receptors versus the next steps in the signal transduction pathway may account for this difference. I'm not going to ask you those sorts of questions on an exam. I'm going to ask you the simple case where you don't need to worry about those sorts of things. We only need to worry about the actual function of the receptors and how they interact with the next step.
Wait, in the last slide, we said that we need to turn off all 5 to enable the response. But here we're saying that we need to communicate 3.
Right.
Doesn't it seem contradictory?
Well, that's a good question. Perhaps you should think about that. Perhaps there's the possibility it could show up in an exam. Yes. Yeah, you should be able to think about that. I mean, let me. If you haven't, you probably haven't looked at the responses that I gave to you to your reading questions. For most of the responses, I didn't answer them for you. I gave you things you should be thinking about to get your way to the answer. The same thing here, right? We've talked about all the stuff that you need to answer the question that Hemi just suggested, right? So I want you to think this stuff through. Talk about it among yourselves. I don't care. The most important thing is that you convince yourself that you can explain it. Okay. Not going to spend any time on this, but one of the things that we've talked about before, and it comes up again, that the receptors for ethylene, the ethylene receptor was the first one for plant hormones that was discovered, and it was discovered because of its similarity to bacterial two-component systems, right? So we see a similar structure of the proteins in terms of functional domains in the ethylene receptor as we see in the bacterial two-component system. We also saw that in cytokinins. I think it was cytokinins that we were talking about, that it was the same. Okay, let's see. The book talks about the ethylene signal transduction pathway. Story is the same. The five pieces of a 1,000-piece jigsaw puzzle. So we know that mutations in this protein, clearly a membrane-bound protein, affect the ethylene signaling. We don't even know what membrane it's in, let alone what the protein is doing, right?
Wait, doesn't it show it on the nuclear membrane?
No, they don't know what membrane it's in. I mean, where's the ethylene receptor? Does it tell you right there? It's in the ER, right? But what membrane this protein is in, we don't know, right? And we do know something about these guys. Who remembers? How do we distinguish these two different transcription factors that are part of this ethylene response pathway? What would be the terminology we'd use for these? You should be able to guess this even if you don't know for sure. Early, late, primary, secondary, right? So Ein3 is the main protein that is transcribed directly in response to ethylene. Once this accumulates sufficiently, it produces this other transcription factor, which turns on a whole suite of ethylene-dependent genes. Okay? So it's no different than the models we've talked about in other signaling pathways.
Wait, is Ein3 there already?
what's the, all the machinery for the signal transduction pathway. Is this transcription factor there if it's an early gene? No. No. Say that again? But these must be there at the time the signal arrives, otherwise you can never do this down here, right? So these are not early genes. These are genes that have been expressed under developmental control. This type of cell needs to be able to respond to ethylene, so developmentally it produces these signaling components, right? But it's not until ethylene comes along that these guys are. Okay? Make sure you understand that distinction, right? When we talk about early and late genes, those are genes whose transcription is turned on or turned off by ethylene. Are these genes turned on or turned off by ethylene? Well, it actually turns out that there are some cases where the ethylene receptor is turned on by ethylene. What would we call that? There are some cases where the ethylene response, what do you call this? Receptor is turned on. What kind of physiological response would we call that?
Wait, the receptor or the
receptor.
So there's no pathway.
So is that gonna turn on or turn off the cell's response to ethylene? Turn on, right? So when something turns itself on, what do we call that? Yeah, or in terms of feedback, positive feedback, right? So let's just get to it. We're almost done. Where does positive feedback play an important role in ethylene responses? What physiological responses to ethylene utilize positive feedback? Where ethylene turns on the receptor proteins, where ethylene turns on the genes involved in ethylene biosynthesis. Where does that happen? What physiological response? Ripening, right. Why? What's different about ripening compared to virtually all the other physiological processes we've talked about?
It's more like a non-reversal. You want to keep doing it until it's finished.
Yeah, okay, so what's the end point?
Like all the tissue is gone. And then
Yeah, right. So it's sort of, in a sense, it's a dead end, right? You want to ripen that fruit up, and then nothing happens after that. It gets eaten, right? So the positive response here is to trigger something where once it's triggered, there's no recovery from it. No physiological recovery from it, right? And because ethylene diffuses away, it's not particularly surprising that the ripening response is one that has a positive feedback. Would this be true for all ethylene responses? So give me an example of an ethylene response that wouldn't have a positive feedback. Is leaf senescence a reversible or irreversible process? Would you expect leaf senescence to be positive or negative feedback? Positive feedback, yeah. What's an example that's where you might expect negative feedback?
Triple response.
Triple response, yep. So triple response is reversible. Wounding responses, right? You don't want the wound response to be on forever. So let's just in the last minute here. Let's just talk about climacteric responses in ripening fruits. So I skipped past it, but here's a list of fruits that ripen using ethylene, fruits that ripen not using ethylene. And the thing that distinguishes them is this idea of climacteric response. And that climacteric response is associated with an ethylene-dependent big increase in respiration. What is that increase in respiration accomplishing? It's not a permanent increase in respiration, but it lasts for several days. What's it accomplishing? Yeah, it's making the stuff that's needed to ripen the fruit, to soften the fruit up, to produce the compounds that attract the animals to come and eat it. Okay, so how about a watermelon? How about grapes? Citrus? Do they have seeds in them that are dispersed by animals eating them? Grapes certainly do. I don't know about watermelons. Strawberries certainly do. The reason I'm pointing this out is because I want you to recognize that the explanations that you see in the textbook, seeds drying down versus not drying down, seeds that are dispersed by animals that eat them versus not, that is not across the board, a 0-1 thing. This explanation is not as good as you'd like it to be. Right? So what makes a climacteric versus a non-climacteric fruit is not cut and dry. The physiology is a lot more complicated than that. Okay, let's quit there. The other thing I was going to say something about is leaf senescence, and it's described well in the textbook, and you should have the physiological context to be able to interpret what's going on in leaf senescence now. Okay? So next Tuesday we'll do abscisic acid. Thursday we'll do brassinosteroids, and then we're done with hormones. Then the last week of classes we're only going to do flowering and then plant stress responses. Plant stress responses is a really nice topic to sort of bring all this stuff back together again. Then we're done.